toilet. I need to pee. Is this toilet? No, this is shoes. This toilet? No, this is not. Oh, this toilet, okay.
2 hours later.
Do you know how the urea being excreted from our body? Let's find out. The urine being excreted from our body through the urea cycle. Urea cycle has another name which is Krebs-Henseleit cycle. The definition of urea cycle is to transform a toxic byproduct of amino acid metabolism, namely ammonia, into less toxic form urea that can be mobilized to kidney where it is excreted via urine. So basically, the amino acid metabolism will produce the free ammonia. However, the free ammonia is toxic and it cannot be excreted properly by the body. So it needs to be converted to urea, which is non-toxic and can be easily excreted in urine. The conversion of the free ammonia into the urea is what we call urea cycle.
This is the overall urea cycle with the structural formula, and this is the simplified one. Steps in the urea cycle. The first and second steps occur in the mitochondrial matrix, while the third, fourth, and fifth steps occur in the cytosol. The first step is the formation of carbamoyl phosphate. The carbon dioxide, which exists in the body in the form of bicarbonate ion, will react with the free ammonia. This reaction will assist by enzyme carbamoyl phosphate synthetase 1. The product is the carbamoyl phosphate. This step involves 2 ATP. The second step is the formation of citrulline. The carbamoyl phosphate produced earlier will combine with ornithine, which we obtain from the cytoplasm of the step 5. The reaction will produce citrulline and catalyzed by ornithine transcarbamylase, and the citrulline will then transported out to the cytosol. The third step is the synthesis of arginosuccinate. The citrulline will now react with aspartate to form arginosuccinate. This will catalyze by arginosuccinate synthetase using 1 ATP. The fourth step is the cleavage of arginosuccinate. The enzyme arginosuccinate lyase cleaves the arginosuccinate to form fumarate and arginine. The last step is the cleavage of arginine. Arginase enzyme cleaves arginine with the presence of H2O to form urea and ornithine. The ornithine is recycled back into mitochondrial matrix to ensure the continuous cycle of the urea cycle. The first nitrogen of the urea is obtained from the free ammonia of step 1. And the second nitrogen is obtained from the aspartate of step 3. And the carbon is obtained from bicarbonate ion of step 1. And lastly, the oxygen is obtained from H2O of the step 5. The urea produced will dissolve into the blood, which will then transported into the kidney, and lastly excreted in the form of urine.
So this is the overall stoichiometry of the urea cycle. It requires the aspartate to react with the ammonia, carbon dioxide, 3 ATP, and water to produce urea, fumarate, 2 ADP, AMP, 2 phosphate group, and pyrophosphate. This equation is irreversible because it requires the 4 high-energy phosphate bonds are consumed in the synthesis of each molecule of urea. If the urea is not excreted properly, it will be accumulated in our body and can cause diseases to our body, such as hyperammonemia.